watching Hamilton today?
Yeah. 7 o'clock.
And we are live. Welcome back, everybody. Today's going to be pretty fun. You got me today. You have Esther next week, I think. But today we are going to learn how to analyze the plasmids. So make sure that the plasmid purifications you did work. So then when you send stuff off for DNA sequencing, you don't send off nothing. And we'll learn how to send stuff off for DNA sequencing. Figure that out. Sound good? All right, let's get it on. So the first thing we're going to do is we're going to learn a little bit about gel electrophoresis and how it works. So once you know how gel electrophoresis works, the rest is pretty easy. All right, so I'm going to take the camera and we're going to walk over to the lab bench and we're going to do everything over there. Remember, no food or drink at the lab bench. All right, let's see if we got a good view here. How is this looking? Man, the delay on this thing is killer. I can't even see what's going on. Should have some sort of live stream at the same time. Let's pull it down. Maybe I can. Let's see. What if I do it this way? That looks kind of better. It's not stable. Oh gosh. All right. Let's see what we got. All right. All right. There's my setup. Now we're looking good. All right. Okay. So you should have received some tin foil, some alligator clips, and a 9-volt battery. Now, and this Tupperware container. That should be basically all you need to do gel electrophoresis and some sort of buffer. So generally what you use in molecular biology is TAE buffer or something, another running buffer, but theoretically inside the thing you can use anything that has electrolytes. But in order to make the DNA gel turn out good, what you really want is you want to use the buffers that have been figured out to work with molecular biology, this being TAE. So first thing you want to do is mix up some TAE. It is 5 grams per liter, right? So you should have received something like this. TAE, tris EDTA, 5 grams per liter. Measure out 5 grams, put it in 1 liter of water. It's better to use tap water. Distilled water doesn't really have those chemicals and electrolytes, so the electrical current can't run through it as well. Tap water has more of these electrolytes and ions that help the current run through the liquid. So basically what we're trying to do is we're trying to run a current from one side of this box to the other side of this box, right? What we're going to do is we're going to place DNA gel in the middle of the box, and so the current should run through and the DNA should move through the gel and it should separate out. Now, the gel works kind of like a sieve. like a molecular sieve. So the bigger DNA molecules move slower, right? Because they'll bump into more things as they go along. Whereas the smaller DNA molecules will move faster because they won't run into as many things. Now, all we have is we sent you as a 9-volt battery. It's not a lot. Generally, you run electrophoresis at 100 volts or higher. We're only using 9 volts. But if you have other 9-volt batteries, you can connect them in series. So one cool thing that you can do hold on, let me find another 9-volt battery. So one cool thing you could do is you can just connect 9-volt batteries in series. So you could plug the negative end into the positive end like this, and you got 18 volts right there, right? You can connect multiple ones of them like that, and you could, get a good amount of voltage out of it. Or if you have a power supply or something else, DC works DC works just fine for this, right? So any DC voltage that you can put through should work. AC voltage won't work because it's alternating current, right? So that means it goes positive and negative, so the DNA won't go in one direction. So make sure you're always using a DC voltage or some sort of rectified voltage when this is going on. All right, so in order to run the current through, we need something to run the current through the liquid. That's what the pieces of aluminum foil are for. Now, generally, in a normal gel box, what you'll see is you'll see platinum wire, right? But instead of platinum wire, so platinum wire won't degrade. Instead, we'll use aluminum. Now, aluminum will degrade over time, so you're going to have to change it out, and it will leave a residue inside your gel, so you're going to have to be careful about that, right? But you can use aluminum, reuse aluminum. It's really inexpensive, and it's really great to use as an electrode for the gel box. Now, when you're working with these voltages, remember not to connect a circuit, right? So when these things are plugged in, don't touch the 2 electrodes at once. Don't stick 2 fingers into the liquid at once or anything like that. You'll get a shock for sure. It's happened to me, right? So be careful. You are working with electricity and you can shock yourself, right? So what you should do is once you've created your TAE buffer, take your TAE buffer and dump it into your Tupperware container. After that, take your piece of aluminum foil and stick it down so the end is in and touching the bottom and the top comes out on both sides. And then what you want to do is you want to connect your alligator clip to the end of this. Don't put your alligator clip inside the liquid because what will happen is that will start to degrade from the electrolysis reaction that's going on. Right? Once we have this, this is basically a functioning gel box. So what you want to do is on the sides of the 9-volt battery, it has positive and negative ends. If you're using a power supply, right, the red is the positive and black is the negative. So what you want to do is you always want to go black to red. If you have gloves on, you will not get shocked. You shouldn't. If you have rubber gloves on or anything like that. Good idea. Wear rubber gloves when you do this. So what you want to do is you want to go black to red, right? Just think of like black to red or else your gel's dead. Black to red or else your DNA won't get fed. I don't know. Think of something, but remember that your gel should run from black to red. So we like to put the negative or black side on the left and the positive or red side on the right, right? So what you can do is you can do a little test. We can connect the red to the positive and the black to the negative. And as the current runs through the gel, what you should see, if it happens, if this battery is not dead, you should see bubbles start to form on your electrodes. Bubbles starting to form on your electrodes is an indication that this is working. They're hard to see right now with this thing. What I can do is I can give a higher voltage and hopefully we can see them better. Let's try connecting two 9-volt batteries. See what happens. See if it's more obvious. All right. Oh yeah, I could start to see them. Let's see. It might be hard to see on the camera, but you should start to see little bubbles form and little bubbles come off. The higher the voltage, the more bubbles you'll see. So let's see if I connect it to, I have an actual power supply over here. Let's connect it so maybe you can see what it looks like. All right. All right. All right, this is becoming too complicated. All right, trust me when I say that there should be bubbles coming out. You should see bubbles. So when you do this yourself, look and you should see bubbles. Yes, so the DNA will migrate in relation to the voltage and current that's being applied across the gel, right? So the more voltage and current, the faster the DNA is going to move. So the slower the DNA is going to move. So when using a 9-volt battery, it's probably going to take you somewhere around 30 minutes, 30 minutes to 45 minutes for the DNA to move into the gel. All right? So the next thing we're going to do is we're going to create a gel. So you all should have received a gel tray, something like this. Now, in order to make a gel, what you have to do is we have to seal up the ends. Now, what you need is some tape. Duct tape, some kind of tape. I know this sounds crazy, but it actually works. So what you're going to do is take tape and line up the top of the tape with the top of the gel and wrap it around the sides. So fold over the tape along the bottom of the gel tray and then make sure you push it down really good and you smooth it out. Make sure this gel tray, make sure you go around the edges and it's pushed down really well so the gel doesn't leak out. But that should be good. Do it on both sides. All right, so let's take some tape and then let's put the tape on the other side. Fold it around, push it down, make sure we got it down nice and good. I like to rub it against the table, make sure we smooth it out on all the sides, and boom, we got a gel tray that's ready to put gel in it. So the next thing you got to do is you got to make your gel. So in order to do that, what we are going to do is we are going to weigh out Agarose. So get your Agarose, and we're going to weigh out, let's say, so we're gonna be looking at plasmids. Plasmids are really big pieces of DNA. So normally you run a 0.5% to 1% gel. So let's do around 0.3 grams of Agarose. And we're going to put about 60 mLs of TAE in here. So in order to measure 60 mLs of TAE, take a tube. You should have a 50 ml tube or 15 ml tube that has gradations on it, right? So take that. And let's dump in. That's 50 mLs of TAE. Let's dump in 10 more. Then we have 60 mLs of TAE right there. All right. Now the next step is you got to heat it up in the microwave, right? Don't put the lid on completely tight. You don't worry if you have a different glass bottle. Now, when you're heating it up, once it starts to boil, stop and let it cool down for a little bit. I know some people have problems heating stuff up. It boils over. You don't have to keep heating it up. Once you start heating it up, stop, let it cool down, and heat it back up. What it does is it'll keep the temperature at a constant high so that everything can melt, right? So what we're going to do, go over, got a microwave. Because nothing's hazardous in here, you can use, you know, your normal microwave, but it's also good to eventually get a microwave dedicated to just lab work. You can get microwaves pretty cheap, especially on Craigslist. So I know it's about 45 seconds to a minute to heat this stuff up. So we will put it in for 45 seconds a minute, and we'll let it heat up. Sound is gone. Can you hear me? Is the sound not working? Hello? No sound? Or yes sound? All right. So microwave's over. And so if it looks like this, there's still some crystals, little crystal things floating in there. It means it's not completely dissolved. So what we can do is heat it up for another 15, 20 seconds. Also, it'll eventually dissolve if you leave it there because it's already hot. So you just have, it takes more time if you leave it there. All right, so we heat it up for a little bit longer. Make sure you're wearing gloves or protect yourself. All right. So it looks like most of the stuff is dissolved. And so what we're going to do now is we are going to add. Yes, correct. The bigger the DNA, the lower the gel concentration. It makes the DNA easier to move through the gel. All right. So what we're going to do now is we are going to pour our gel. All right, so here's our gel tray. So what we want to do is we want to put a comb. These things are combs. So each one of these is going to create a well in our gel that we can add DNA or other things to. And so if you have your plasmids, you probably have 3 samples, plus you want 1 sample for your ladder. And if you have a control or something like that, another sample. So what you'll have is maybe 5 samples. If you want, you can use 2 combs if you have a lot of samples. But in this case, we're only going to use 1. So just put 1 comb. There's little notches near the top. It doesn't have to go all the way down into the notch. It's okay if it just rests on top of the tape, right? The next thing we're going to have to do is we're going to have to add some some DNA dye to our gel. So what you want to do is you want to take your 0.5 ml, 0.5 microliter to 10 microliter pipette. And what we're going to do is we're going to add 5 microliters to our solution so that we could visualize our DNA. Oh, yeah. So take your DNA dye, take 5 microliters of it, 5 microliters of your DNA dye, and just pipette it directly into your molten agarose. Actually, because we did a 60 ml, you probably want to do 6 microliters, but it doesn't matter too much. You still should be able to see it. So the DNA dye is 1 to 10,000. So what we're going to do is we're going to take our agarose solution and we're going to dump it into our gel tray. All right. Then what we're going to do is we're going to take our gel tray and stick it in the fridge. Sticking it in the fridge helps it cool down faster and make sure that it doesn't leak because the agarose will solidify faster. I'm just going to go stick it in the fridge. All right, now that should be ready in like 10 minutes. In the meantime, what we need to do is we need to get our samples ready. All right, so you should have received some parafilm. Now parafilm is great for many things, but what it has a very like hydrophobic surface. So that surface allows you to put liquids on it, and you know, how is it not boiling over? Mine always tries to boil over, and I have to do it in 3 to 5. McKinley. Just you must have a really powerful microwave. So yeah, just do it really slow, right? When it boils over, just let it sit for a little while. Don't let it boil over. If it's boiling too much, just let it. You don't have to keep heating it. Your microwave is probably just really powerful, right? So we have our parafilm here. So the things you're going to need your 1 kb DNA ladder, and you're going to need your DNA loading buffer. You're going to need your plasmids that you want to look at. So I got 2 plasmids here that I'm going to look at. And we're going to run these out on the gel when the gel's ready. So what we're going to do is, in order to put let's see if I can get a close-up on this. How's that looking? This is difficult. My bench is really messy, so sorry. Gonna have to clean this up a little bit. Let's see what we can do. Let's clean this thing up. All right. All right. Okay. All right. There we go. All right. So here's the piece of parafilm, right? So what you're going to want to do, we have a DNA loading dye that is 6x. So what we're going to want to do is load 10 microliters of each plasmid. But we want to mix the plasmid and the loading dye, because the plasmid DNA itself won't go into the well in the gel, right? But these loading dyes, what they have is they have glycerol. You can use glycerol or you can use sugar. You need something to mix with the DNA that will weigh it down, right? It's like when you add something that's more viscous to a less viscous solution, the thing goes to the bottom. That's what we want. We want this solution, the DNA, to go to the bottom of the well. So in order to mix them, you don't need to do this. A trick scientists do is you could take 3 or 4 microliters, even though it's 6x, and pipette little drops onto the parafilm. Now what this does is it allows us to easily mix the DNA with the drops, right? So now what we can do is we can take 10 microliters of DNA and mix it by just pipetting up and down in the drop. Voila, there we go. And that we have a nice solution we can pipette into our gel. I don't know if it's really in focus well, but hopefully you get the idea. All right. So now we're going to do that with all the plasmids that you have and get ready to run a gel. All right? You're going to get ready to run the gel. So once our gel is solidified, we're going to add the ladder, we're going to add our plasmids, and then we're going to run it. All right? So any questions while yeah. You know, I have a GoPro, but it's like you can't literally stream with the GoPro, right? At least I don't know how to stream directly with the GoPro. So what you need is you need like some complicated machinery to stream with the GoPro and also do this stuff at the same time. All right. All right. So we got about 10 minutes to burn. So what I'm going to do is I'm going to teach you is the film used industrially. Parafilm, I don't know. I don't know if it's mostly used in science or if it's also used in other things industrially or things like that. All right. So while we wait for our gel to solidify, I'm going to teach you how to order DNA sequencing. All right. So let me see if I can get the window I want on here. GeneWiz, Google Chrome. I think I have too many Google Chrome windows open is the problem. Let's see. Window capture. Okay. Not the bioengineering class. Not that. Let's see. No. Gosh darn it. All right. Let me see if I can log in on Firefox. Oh, right, my laptop is making funky noises. All right, I think I got it. Let's see if we can pull it up on the stream. Yes. Okay. Let me change the size. All right. So what you're going to want to do, first thing you want to do is sign up for GeneWiz if you're in the US and log in. I don't know if you can use it if you're outside the US. You might be able to, but what you can do is you can just search for DNA sequencing or Sanger DNA sequencing. So Sanger, you can see right here, Sanger sequencing, and find a company in your country that you can do this. We will also be providing example data for people who haven't got around to sequencing or can't find a company in their country yet that will sequence, right? So when we submit something for sequencing, so sign up for the website, log in, go to the home, services and products, and we want to do Sanger sequencing. Now there's all these different things for Sanger sequencing, all these different things you can do, but what we're going to do is plasmid. We're working with plasmids, obviously, and this is how we'll get to sequencing what we want to sequence, right? So for this, because we want GeneWiz to use a primer that they already have, we are going to go predefined. So if you're doing other different types of sequencing, you might use premix or custom or these other things. But for our cases in this class, we're going to use predefined, right? And depending on how many samples you have, 1, 2, or 3, so how many plasmids you have, you're going to want to put in that many samples. Now we go down here. So this is the name that you're going to label your tube as. You could put the name as anything, right? So what do you want? So we have a GFP and we have a P dusk and we have a P dawn. So you can use those 3. So the length in base pairs, I never know if this is actual length of the plasmid or the length of the sequence you want. I don't think it matters so much. The length of the sequence we want is, somewhere between 500 and 1,000. You click this, it'll fill all values. The concentration, put in. Now we'll figure out a little trick when we run the gel on how to calculate general estimates of concentration. This doesn't matter too much. So a good estimate from a mini prep is 50 to 100 nanograms per microliter. So just put 50 nanograms per microliter. Now we don't have a primer, but we're going to use a GeneWiz primer. So GeneWiz tells you what, oh, we don't want to click on that. GeneWiz tells you, all right, what primer do you want to use? So in this case, we click on here and we scroll down. What primer we want to use is the T7 term primer. So one cool thing is, if you remember from our first lecture, let's see, let's find P dusk maybe. Map. So you can see in P dusk plasmid it has this T7 terminator. This is what the T7 term primer binds to. And so what sequencing does is you'll have a piece of DNA that binds to the piece of DNA that you send in, and this piece of DNA, slowly nucleotides will be added to it. And as the nucleotides are added, it'll give a signal, right? And this signal will tell them what sequence it is. So slowly, if we use the T7 terminator, it should walk backward across the sequence. It should be able to tell us what all these DNAs are in our plasmid, and it should help us identify if we have the correct plasmid, number 1, and if what sequence we have in here. We know what sequence it is, but we're just doing this to learn how to do sequencing. So all the plasmids have this T7 term. You can look in the plasmid map. So we're going to put all that in. Now, once you have this, you're pretty much done. So what you're going to want to do is go to Save and Review. Primer source GeneWiz primer, right? We want GeneWiz to use their own primer. And then add to cart. Now, it costs $7 to submit these sequences each, plus shipping. It's pretty cheap. And what you can do is you can select ship your samples to GeneWiz. It'll give you an address where you ship your samples. We included some bubble mailer envelopes with your kits that you can mail the DNA sequences in. So just take those bubble mailers, write the address on it, take it to the post office, and ship it to GeneWiz, right? Print off a copy of the invoice, or write the invoice number that you receive once you make your order, right, on the package. So there should be a tracking number right here. So include that number in there so they know what samples they have, and you're good to go. Sequencing is that easy. It's that simple. That's all you have to do. They'll notify you when they receive the sequences, and they'll notify you when they finish sequencing, and they'll give you the data to analyze. Next week, we're going to go over how to analyze the DNA sequences. So, you know, if you can submit some stuff this week, then next week you can play around with the DNA sequences. Yes, the ladder is good to go. You don't have to add the solution to the Tupperware at the start, Chris. So the TAE buffer that I added to the start, yeah, you just send the centrifuge tube inside the classroom, the classwork. There should be a document called submitting samples for DNA sequencing or something like that. You don't have to send the whole centrifuge tube. What you can just send is like 20 microliters and save some for yourself, right? So you want to save some DNA for yourself, so don't send it all. Just send 10 to 20 microliters, enough for them to do a DNA sequencing run or two. But that should be it, right? So we send this off, and yeah, we're good to go. All right. Okay, so let's go check on the gel. All right, the gel's almost done. It needs like 2 more minutes or something like that, less than 5 minutes. So anybody have any questions? Yeah, it's okay if it's not refrigerated. So you can send the stuff in a microcentrifuge tube. The DNA shouldn't degrade. So DNA can actually last a pretty long time at room temperature. As long as it's not really dirty. But DNA that you did like a mini prep, plasmid prep on, the DNA should be pretty clean. So it should be able to last a good amount of time at room temperature. So don't worry too much about it being at room temperature and it being refrigerated and stuff like that. Your plasmids that you purify, generally for any sort of long-term storage, you want to store them in the freezer. This is just, you know, safe precautions. It's okay if you store them in the fridge also, but you want to be safe when you're doing molecular biology. You're fighting against probabilities, you know, you're fighting against the probability of making things go right. So generally, yeah, the DNA dye is undiluted. When you mix it with the sample, it becomes diluted, right? So you're diluting it from 6x to 1x when you add the sample. So that is what you're going for. So don't dilute it beforehand. It gets diluted in the sample. Yeah, Jeff, So what we're trying to do here is this is kind of like a practice run. Whenever you get plasmids, you want to sequence them so that you know you have the correct plasmid before you try to do any genetic engineering, right? Because if you don't have the correct plasmid, then there's no point in going forward, right? So what you want to do is you want to verify that you have the correct plasmid before you do anything else. Otherwise, you're just going to waste a lot of time and energy. Because basically, next week we're going to learn how to analyze DNA sequences. And then for the last 3 or 4 weeks, what we're going to do is we're going to do genetic engineering, right? So we're going to take all the things we learned in the first half of the class, plasmid purification, DNA sequencing. We're going to learn some new things like restriction digest and DNA ligation to move pieces of the plasmids around. But we're going to try to use all the things we learned in the first half of class and apply them in the second half of the class, right? So that's the goal. So this is just, this DNA sequencing is a lead up to the other experiments. If you want, you can only send off 1 plasmid or 2 plasmids or whatever if you don't want to spend the $21. But it's really not that much money generally to do DNA sequencing. So any other questions? Come on, we need some questions so we burn a couple more minutes so the gel solidifies. Anything. Give me some questions. You can probably send, so let's look at GeneWiz. So some companies will do that. I don't know if GeneWiz does that. You might be able to contact them and ask them if they will do that for you. They might. But yeah, some companies will do that for you. You're just probably going to have to contact them and ask them. We'll be learning about double-stranded RNA. I don't think we will be talking about double-stranded RNA here in this class. We're mostly just going to be focusing on DNA and what's going on with the DNA. All right, going to check on the gel. All right. So the gel is ready. It has started to solidify. So what we're going to do in this case, in order to get this gel going fast, I'm going to run it using a standard power supply. So these power supplies, you might be able to find some on eBay for like 60 or 70 or 80 bucks. We didn't want to send these because we didn't want to increase the cost of the class. If you're interested in buying power supply from us, we can probably get them for like $75 or something like that. Contact us and we'll try to get you one. They're hard to come by sometimes, but we might be able to find them. All right. So what we are going to do here is so what you want to do is you want to take your TAE buffer and dump it in your gel. You dump it in your box. So instead of using the Tupperware container right now, I'm just going to use a gel box we have. These gel boxes are really difficult to come by. which is crazy because they're just pieces of plastic with some platinum wire. We've been trying to find a cheap, inexpensive person to manufacture them for us, but it's been difficult. Everybody wants to charge a buttload. What you want to do is gently just push your gel out of the tray and into the solution. You want to have your gel completely covered in buffer. So just dump the buffer in until it covers the top of your gel. All right. So now, generally what you do is in the first lane, you're going to want to add your ladder. That's just so you can keep track of your ladder. So depending on what's going on, you're going to want to add 5 to 10 microliters of your ladder. That depends on a lot how well you're seeing it. So if you run your ladder out and you can't see anything, just add a little bit more. I generally add around 5 or 6 microliters. So this is the hard part. Now, adding the liquid to your gel well is hard. It's hard to see the wells in the gel that you're looking for. Now, a trick that you can use is you can take a piece of paper or something that has some color or something like that to it, and you can stick it underneath your box. Now, what this gives is it gives a little bit of contrast on there to be able to see it, right? Because I have lots of experience with this, I kind of know and I can feel. So you want to be really gentle when you do this. So you're going to want to take your pipette and you're going to want to find the well. So you have to be really gentle and kind of just feel around. You know, move your pipette tip, and if you move your pipette tip back and forth and the gel moves back and forth, you know you're in the well. You don't want to stab down with your pipette. If you stab down with your pipette, what you're going to do is you're going to puncture the gel and you're not going to get anything in there. So try to find where the well is, stick your pipette tip in the well really gently and slowly and move it around. Then what you're going to want to do is slowly eject the solution into the well. And what you should see, let's see if you can see this over here. You should see the purple color inside the gel, and it should just stay there just like that, right? So what we're going to do is we're going to do that for all of our samples. So that was our ladder. So next we're going to do for the thing we pipetted already, the plasmid and the DNA dye. We're going to take that. We're going to find the little well in the gel. And we're going to slowly push the stuff in there. And then we're going to take our other plasmid. We're going to mix it with the loading dye. Put it on the parafilm, mix it in and out. And we're going to slowly put that in there. All right. So now what we have, you can see, we have our three lanes loaded. Let's see if you can see the wells. No, it's pretty hard to see the wells on the camera. All right. So now when you have your 3 lanes loaded, what you're going to do is you're going to attach the electrodes to the battery or power supply or whatever you have and run it. In this case, we're going to use a gel electrophoresis power supply just so we could run it at high voltage and get this gel run quicker. So the higher the voltage and current, the quicker you can get it to run. I know, this is a mess. So connect the electrodes. Let's run it really high. So we'll run fast. We'll run it at like 100. So what you could see here is you should be able to see the bubbles coming up. Now these bubbles, can you see the bubbles? Kind of, not really. The focus isn't really good on this thing. Yeah, there you can kind of see the bubbles streaming up from the electrode. All right, anyway, all right, so now what we're going to do is these DNAs should move from the black, so from the negative electrode to the positive electrode, so black to red. So we should slowly start to see these buffer, so you should start to see the dye and the DNA move, right? So you should start to see the purple slowly move down the gel. Now in the case that we're running it really fast like this, it should happen in a few minutes. With lower voltage and less, it's probably going to take a little bit longer. All right, so let's give that a few minutes. So now what we should be waiting to see is if, number one, if we purified our DNA and what concentration our DNA is. We can estimate the concentration of the DNA. So you can reuse the gel multiple times. What you do after you're finished with the gel is just break it back up and either save it in a Ziploc bag, stick it in the fridge, or you can just put it back in the glass bottle and seal the glass bottle. Then all you have to do is reheat it, right? So when you reheat the gel, what happens is that all the DNA will just spread everywhere, right? So the DNA gets mixed in with the gel, and so you can reuse it without any issues. You can reuse it probably, I would say, I would use it at most about 3 times. If you're really desperate, you might be able to get more. The problem is that the buffer in the gel starts to get electrolyzed, so it doesn't conduct current as well. And then what ends up happening is the DNA doesn't move well through the gel and it looks all messed up and distorted. Pamela, if you have a power supply, what should the setting be? So generally what you run at is somewhere around 150 volts and 250 milliamps. If you just have a voltage setting, just set it at 150 volts. And you'll probably run that for 100, you'll probably run that for like 15 or 20 minutes. Maybe less. You can see what's going on your gel just by looking at it. So you can tell like how far along the stuff is moving and how fast it's moving, right? But generally, like I said, with any DC voltage you could run through the gel, it should move the DNA. Jeff, if we have a PCR machine, does that mean you can't really make copies of plasmids with a PCR machine? You kind of can, but why don't I just 3D print a mold and vacuum form gel boxes? Why don't I just do that? Well, it doesn't sound like I could just do that. It sounds like it's going to take time and money. Unfortunately, running a business, you don't have a lot of time or money. But hey, man, if you want to 3D print a mold and vacuum form gel boxes for us, we'll buy them off you. So, all right, so let's go check on that gel, see how it's doing. So what we can see, it might be hard to see, is that the DNA has started to move outside the wells and down into the gel. Now, I'm going to try to see if we can visualize this using our camera. So let me pause this gel. This is going to be difficult. So what I'm going to do, you all should receive an orange pair of glasses and a blue light. Now, what that does is the glasses help block out the light so you can see the fluorescence of something fluorescing. Now, in this case, what we're looking for fluorescing is DNA, right? So trying to find out where some glasses are. I don't need glasses necessarily. I have this green thing. Ooh, this is going to be difficult to see. Let me turn off the lights in here. Okay, let's see if we can see this. It might be complicated. So let's put the filter in front of it. Hold it close to the gel. And we're going to shine. Oh, you see those little green band things? Let me see if I can make this more diffuse. Alright. No, it's not working well. It's really hard to see on here. Alright, so what we should be seeing here is, let me get a picture. Let me pull up a picture. So, alright. So what you should see is something that looks kind of like this, right? So you should see these green, when you have on the orange glasses and you're shining the blue light on the gel, you should see these little bands that look green, and they should look like this, right? Now, if you successfully purified plasmid DNA, you should see a band like this. And the band should be running above all the ladder, most likely. That's because the DNA is pretty big, and the way the DNA is formed on the gel is going to run really high. So what you're looking for is, if you successfully purify DNA in your mini preps, you should see something that's running in the lane that you loaded, right? So our ladder should look something like this, and our plasmid lane should look something like this. Should be some band like this. Now, if that happens, you know that you successfully did a mini prep. Sometimes you successfully did it, the concentration is just too low. So what you can do is you can take your gel, melt it, add another 5 microliters of the DNA dye, right? So if you add more dye, it'll help you be able to see stuff better. And try to run the gel with some plasma DNA again. The other thing, if you're having trouble seeing if there's any DNA or bands, turn off all the lights, right? Go into a room that's dark and look in there. Look at the gel in a dark room. If you have a camera that does a long exposure, take a long exposure picture of it. That can help you see the DNA bands better. So all these things will help you see it, but it should look something like this. Now, the way that you can estimate the DNA, the quantity of DNA, is generally, you can see that some of these bands on the gel are brighter, on the ladder are brighter than others, right? So when you run a DNA ladder, you'll see that some bands are brighter than others. Now, the 1 KB ladder we use at the ODIN, so we can go to the website, we can go 1 KB, 1 kb DNA ladder. So the brighter bands have 100 nanograms of DNA, and the other bands have about 50 nanograms of DNA, right? So the 4,000 base pair band has 100 nanograms of DNA. So what we can do is if we look back on a gel, right, you can compare your band and its brightness to the band on the gel, the brightness on the band on the gel, and you could estimate approximately how much DNA you might have in your sample, right? So for every approximate size of the band, right? So if we're looking at the 4,000 base pair band, if it's twice as big and bright as the 4,000 base pair band, that means that there's about 200 nanograms of DNA in there, right? If it's about half as bright, then that means there's about 50 nanograms of DNA in there. This is obviously just an estimate and is not an exact science, but you'd be surprised at how well you can actually estimate the amount of DNA in a DNA ladder using this technique, right? So if you see something on your gel for where you did a mini prep, then it's great to send it off for sequencing. Even if you didn't see anything, it's probably still okay to send off for sequencing just to see what happens. It's $7. If you want to save the $7, you know, feel free not to, but it's $7. So sometimes there's actually something in your sample and you just can't see it on a gel or something like that if it's a lower concentration. What you can also do is you can rerun your mini preps, right? So if you're rerunning your mini preps, instead of using one microcentrifuge tube to do your mini prep, what you can do is you can dump out the liquid, add some more, spin it down again, so you can have, you know, 2 or 3 or 4 microcentrifuge tubes of culture that you pelleted that you use for your mini prep. The more bacteria you use, the more DNA you can get, right? So using more bacteria in your mini prep will get you more DNA. If you run out of materials for your mini preps or things like that, just contact us and let us know, and we will send you more. So just, you know, try to get this stuff done, but we will send you more if you run out. Don't worry about that. All right, so yeah, it's a good idea to set a timer when you run your gel so you don't forget about it. Set a timer for like everything you do in molecular biology, because let me tell you, people always say like, ooh, I'm going to tell myself that like, that I'm not going to forget this. I'm going to remember it. I'm not going to forget it. But everybody always forgets it. So like always set a timer. Always set a timer when you're doing stuff. So David says, GeneWiz is asking for nanograms per microliter, whereas that says nanograms per 5 microliters. Yes, that's correct. What we were talking about was total nanograms, right? Or nanograms per 5 microliters. So when you're measuring the brightness, it's total nanograms for the amount you loaded. It's not nanograms per microliter. But the number that you give GeneWiz technically really doesn't matter, right? So generally what's going to happen is your DNA sequencing is going to work or it's not going to work. There's usually not an in-between, and I've sent really low concentrations of DNA and had it work, and really high concentrations of DNA and had it work. So it's not an exact science, the concentration or the amount of DNA that you send, right? Because like I was talking about, you know, this stuff it's all probabilities. So the probability that the sequencing reaction works is dependent on how much DNA you have and the quality of DNA. But it can still work even if you don't have a lot of DNA. But if you don't have a lot of DNA and the quality of DNA is bad, the chances it works is a lot less, right? So your goal is to send a lot of DNA that's really quality, but you can get away with a lot less. So, all right. So, any questions? If you have questions while you're doing this, post your questions in the Google Classroom so then everybody can see. I know some people have been, emailing us and messaging us. It's usually better to post your messages, your questions in the Google Classroom, because if you have the question, other people probably have the same question and it helps everybody to learn. So if you have an emergency, feel free to message us on Facebook and email us, like, oh, I'm doing this right now and I need an answer. But generally try to post your questions on the Google Classroom because it helps everybody out that way. All right. So try to submit some of your samples for DNA sequencing in the next week or two. And next week, next Tuesday, Esther's going to go over with you like how to look at your DNA sequences, how to analyze your DNA sequences, and stuff like that. But even if you don't have any DNA sequencing, we'll provide you with some example sequencing that you can work with. So don't worry about Dave. If you're stingy, send 10 microliters. If you're not stingy, send 20 microliters to GeneWiz. Sometimes I think they generally, if your sequencing doesn't work, they'll run a second reaction for free. So, I generally send, if I don't want to do another mini prep, I'll send 10 microliters. So then you have DNA left over for more experiments. But yeah, there's no exact number you have to send. It just really depends on how comfortable you feel. Like if you're comfortable that the DNA sequence is probably going to work, send them 10 microliters. If you're not, feel free to send them more. It gives them more DNA to work with. They can run the reaction again, stuff like that. Alrighty. All right. We've been going for a bit, so I am going to take off. So hope you all have a good one. I'm going to be in China running some workshops there for the next week. So I will see you all when I get back. But until then, keep at it. I'll probably still be online and stuff like that, so you can contact me there. But Esther and Jill are going to take over for the next week. So next class will be done by Esther and Jill. But I'll be back to class after that. Alrighty.